Okay, sebelum kita mula, saya nak check dulu berapa orang dah masuk pedek. Alright. So, ada 15 orang dalam ni. Semua ni dah makan ke belum ni? Dah breakfast ke? Ashraf dah breakfast. Nurin masuk awal tadi dah sempat breakfast tak Nurin?
Sempat cikgu.
Sempat. Okey, bagus. Okay. So, anak-anak, please take a look at your paddock right now. Sebab kita for the first few parts of our class today, saya nak address beberapa concern. I want to address a few concerns. Sekejap ya. Oh, sudah makan, Fatimah sudah makan. Okay, Alhamdulillah, bagus. Okay, apa yang nak sebut tadi? Okay, dalam a few earlier slides in our paddock today, I would like to address several concerns that you shared during reflection. Reflection tu adalah sangat penting pada saya sebab saya nak tahu part mana yang kamu rasa tak boleh lagi, tak boleh nak proceed lagi, tak boleh nak selesaikan lagi. So, thank you so much for your reflection last week. Based on your reflection, I decided that the first few of our discussion today is applying the concepts that we have learned last week so that you can see what is the context, macam mana kita nak guna apa yang kita dah belajar last week so that you can increase your understanding. Okay, so concern last week ialah mostly pasal facilitated diffusion and active transport. Number one, issue dia ialah the term that we use in our textbook for facilitated diffusion is a bit different compared to the video. Sebab video uses the term channel mediated or carrier protein mediated facilitated diffusion kan. So mungkin ada confusion juga sikit-sikit kat situ. Number one. Number two, ada yang masih belum clear about active transport. Number two. And number three, okay, a few of you also mentioned about not understanding exchange of gases in alveolus, between alveolus and blood capillary. So, to summarize that, those are the three main concerns, macam semua kan, tapi tak adalah semua sebab yang sebut ni the total number of concerns ni yang sebut ada dalam lebih kurang 10 orang. So I hope by addressing the issues, addressing your concerns through assessment like this, I will help to increase your understanding better. So let's take a look at the first slide where you need to label the diagram. Okay, so P is hydrophilic head. Kena tambah head. Hydrophilic is the adjective. So the structure is head. It is also acceptable if you say that it is a polar head and non-polar tail. Atau polar head and non-polar tail. Also, it is also acceptable for you to write hydrophilic head and hydrophobic tail for Q. Okay, sebab you need to add head and tail. For P is hydrophilic head, for Q is hydrophobic tail. You need to add the head and tail because hydrophilic and hydrophobic is the characteristic. It is not the structure. So you need to include hydrophilic head, hydrophobic tail. It is also acceptable for you to write polar head and non-polar tail. Okay, what is the difference between R and S? R is, there is a pore, ada straight line dekat tengah-tengah tu kan. There is a straight channel in the middle. There is a straight pore in the middle. So R is channel protein. And S is carrier protein. Okay, S is carrier protein. That is the structure of carrier protein, the way we draw carrier protein without seeing the binding site. When we see the binding site of carrier protein atau ada certain reference name that as active site. So the binding site of carrier protein, we can see that if we draw a cross section of carrier protein. But the one shown in the diagram as, okay, S is a carrier protein. Okay, S is a carrier protein. Alright. Okay, most of you got it right. Okay, at the center of R is channel atau pore. So some references mention it as pore protein. Okay. So I give you around 30 seconds to update the slide. Saya minta maaf saya menggunakan sistem timer sebab lebih kepada untuk mengingatkan diri saya sendiri supaya tidak spend masa terlalu lama pada sesuatu slide, because certain parts are only recall. Setengah bahagian tu hanyalah untuk menguji kebolehan recall saja, mengingat kembali saja. So tak boleh nak dwell on it too much because when we go through questions such as explaining and describing questions, we will reuse the ability to recall the name of the structures in that particular question. So kita akan recall banyak kali nama struktur-struktur ni ya. Okay, let's take a look. You have already identified P, Q, R and S, where P is polar head, Q is non-polar tail, R is channel protein, S is carrier protein. Can you give one example of a particle that diffuses across Q, diffuses across R, and diffuses across S? And then can you mention the process? Can you mention the process? How the molecule move across structure tu? Nama process. We have discussed 4 different process last week. So what is the name of the process for particle that crosses atau diffuses through Q, particle that diffuses through R, and the particle that diffuses through S. Basically soalan yang lebih kurang sama, but now we have already discussed about different types of transport. So this is where I assess whether you understand or not our discussion last week, and I can re-clarify it today. Okay, we already have 31 here. When give one example, just state the name of the substance. Bagi satu, bukan karakteristik tau. Jangan small, non-polar, charge, itu adalah karakteristik. Give the name of the particle. And then in our cells, when discussing about passive transport, it is very rare. Dalam silibus kamu pun tak ada sebenarnya. We don't talk about transport of atom. Substances tu usually exist as molecules ke, ion ke, jarang sekali dia exist as atom. Misalnya kita tak discuss transport of carbon. Carbon is atom. But we discuss the transport of carbon dioxide, the molecule. Kan? Okay. Lagi kita tak discuss transport of oxygen, but we discuss about transport of oxygen gas. So if you want to write, you either write O2 or you write oxygen gas. Baru betul. Okay. Sebab kita tidak transport oxygen atom. What we transport is oxygen gas atau oxygen molecule O2. Okay. Tak payah label cross section tu dah sebab kita dah label tadi. Okay, very good. Okay, ramai yang dah siap. Okay, diffuse across Q, ramai yang tulis either water or oxygen gas. Siapa yang tulis oxygen ni sayangku, you need to add gas. Okay, kena tulis oxygen gas. Okay, kalau kamu tulis O2, tak tulis gas pun tak apa. Sebab memang it is O2 is the standard way to annotate the oxygen gas. It is a standard way to annotate the oxygen gas. When you write O2, we straight away understand it is a gas. Macam tu ya. Kalau kita tulis O2 saja pun cukup. But if we write it in a full word oxygen, so oxygen only is an atom, but if we write oxygen gas or oxygen molecule, we know that we refer to O2. Okay. So the first one, ramai yang sebut water, ramai yang mention oxygen, dan saya jumpa seorang yang tulis glycerol. Boleh? Tak ada masalah? Betul tu, glycerol. Okay, so if the substance want to go through Q, the molecule move across via simple diffusion. Pandai. Dia dah nampak dah tu, Pandai, simple diffusion. Bagus, that is the first part. Alamak, siapa tingtong tadi ni terlepas. Alright, okay, the next one. Okay, Puteri Nur Azlina, selamat datang. Alright, next is R. R is channel protein kan? Tadi kita label R is channel protein kan? So channel protein, siapa lalu channel protein? Who diffuse across channel protein? Channel protein is for ion. Bagus. Okay, so sama ada tulis Na+, K+, or you write full name sodium ion. Kalau tulis ion saja, tak dapat lagi markah sayangku sebab you are required to write one example. So, one example mesti kata nama khas. Which ion are you referring to? Potassium ion ke? Chloride ion ke? So, siapa yang tulis untuk R, ion saja, betul kan no? If you write sodium saja, pun kena betulkan, kena tambah sodium ion. Hang kalau tak mau tulis panjang-panjang, hang tulis Na+. Lagi senang. Okay. So if you write sodium ion. Okay, good. Facilitated diffusion. Whereas S, dia punya example dia ialah glucose and amino acid. Glucose pun boleh. Amino acid pun boleh. The way for glucose and amino acid to diffuse across is facilitated diffusion. Okay, I can see here one of you wrote for S, partikel dia ialah glycerol. Glycerol boleh dissolve dalam lipid. So, glycerol is an example for Q. Okay, ada seorang jawab glycerol ya kat sini. S ni sayangku, betul kan? Siapa yang jawab contoh S tu glycerol tu? Tukar kepada glukos. Dia tersasul kot tu. Glycerol glukos. Yelah nama nak dekat sama kan? Okay, good. Tulis. Sodium ion. Beza dia jenis transport protein saja kan. Both R and S are transport proteins. So whatever substance that goes through R, that goes to S is is a part of atau move across the plasma membrane through facilitated diffusion. However, if you are ions, you go through R. If you are a big molecule but not charged, tak ada charge, you are neither positive or negative, but you are a big molecule, you have to go through S, which is carrier protein. Okay, so itu pengenalan kepada facilitated diffusion. Now, tadi kita recall label. Tadi kita buat soalan aras pertama, aras rendah, iaitu kita simply recall what we remember. Okay, this one is a bit higher where we relate the name of the the example of particle and the specific name of the mechanism. Let's take a look at the next one. Okay, saya pergi ke next one ya. Next one level tinggi sikit. Explain how the particle you suggested before move across S. Kalau S tadi hang cerita, hang kata glucose, hang tulis how glucose move across S. If previously you said the example is amino acid, explain how amino acid move across S. The mark for this question is 3. Okay, markah untuk soalan ni adalah 3. Gunakan gambar pada sebelah kanan yang saya berikan tu sebagai panduan. Okay, sebagai panduan. Alright. Tulis-tulis tak apa. You write whatever you remember, the step by step process. Lepas ni bila saya tengok, saya akan bantu untuk tambahkan lagi, kemaskinikan lagi cara kamu tulis. So anak-anak, tengok gambar. Okay, if you don't remember, tengok gambar. Name of the substance. If you don't remember, tengok gambar. Okay, tak apa. Kita cuba sama-sama. Okay. Okay. Dia nak explain, dia tak tahu macam mana nak jawab tu, panjang daripada jawapan macam mana nak cerita dari satu persatu. Okey anak-anak, untuk mereka yang tak jawab, tak mula jawab lagi, ramai ni, Ashraf, Fatimah, Alif dan sebagainya ni, meh saya tolong. It's okay, bagus. Dia, thank you for reaching out. Okay, so nombor 1, whenever we talk about, orang yang tengah tulis ni, teruskan menulis je, tak ada masalah. Lepas tu, hang tengok kat situ, saya dah nampak dah ada orang tulis ATP pula. Tengok gambar, ada ATP ke? Tak ada kan pada gambar kanan tu. Why you put ATP when there is no ATP in the figure? Okay, ini untuk orang-orang yang reaching out tadi, dia kata clueless. Okay, untuk orang-orang yang tengah tulis ni, markah ni, soalan ni markah dia 3, tapi points dia ada sampai 5 to 6. So if what you are writing right now, mana-mana 3 point kena mana-mana 6 point yang saya cerita, don't fret ya, jangan risau. Asalkan ada Okay, untuk orang-orang yang tak tahu ni nak tulis apa, hampa gunakan apa yang Allah dah bagi pada hampa, iaitu mata. Okay, gunakan mata hampa, tengok gambar sebelah kanan. I've given to you already. In the original question, gambar kanan ni tak ada You have to recall the process of facilitated diffusion to answer this question. So point number 1, cerita. Dekat mana, okay, usually part yang berwarna ni ialah part yang dalam sel, part yang tak berwarna ni part di luar sel. Kalau kamu nampak warna biru versus warna warna brown ke apa, selalunya warna biru ni outside the cell, warna brown ke apa ni inside the cell. Okay, warna biru, warna putih adalah representing outside the cell. Okay Itu starting. Okay, Now, untuk mereka yang tak tahu nak jawab apa ni, nombor satu, cuba kejap bila kita nak discuss transport of certain substances, first we identify the concentration. Ada tak difference in concentration? That should be your P1. Kenapa itu that should be your P1? Sebab itu yang kita nampak dalam gambar. Okay. So, katakanlah tadi kamu pilih glukosa. Meh cuba. Jangan biar kosong macam ni. Jangan biar kosong. Mesti cuba jawab. Mesti cuba. Okay. So, point number 1. Tengok gambar. Tadi kamu pilih untuk untuk S, kamu pilih glukosa ke amino acid. So, point 1, kamu bagi tahu concentration glucose. Dekat mana tinggi, dekat mana rendah. That should be your point 1. Okay. Oh, sorry. Saya tertukar. Okay, sorry. Okay. Point 1 should be, okay, based on the diagram I've given you, dot merah tu represent concentration. So, point 1. Concentration of glucose. Mana tinggi, mana rendah. Okay. Okay. Macam saya sebut tadi, kawasan yang warna putih tu luar sel, kawasan yang berwarna tu dalam sel. So, kita boleh bagi nama pada region tersebut. So, kita tahu daripada gambar ni, concentration of glucose, sayangku, bandingkan dulu kepekatan glukos. Bandingkan dulu kepekatan glukos. Okay. Siapa yang tinggi, siapa yang rendah? Terus bagi. The concentration of glucose outside the cell is higher than inside the cell. Kenapa saya tahu outside the cell is higher than inside the cell? Warna putih tu biasanya represents, kalau kita tak nampak kawasan tu berwarna, kita tahu kawasan tak berwarna tu adalah luar sel. Yang berwarna tu dalam sel. Sebab blood plasma biasanya tidak berwarna. Okay. So, high concentration of particles tu outside the cell. Now, the concentration of glucose outside the cell is higher compared to inside the cell. Siapa yang tengah jawab tu tak ada masalah. Teruskan jawab. If you know already, kalau ada nak rasa nak tambah, tambah. Okay, jangan risau ya. Bukan jawapan kamu salah. Okay, sebab soalan yang sama boleh push sampai 6 markah. Tapi sekarang ni kita nak minta 3 saja. That is point number 1. Okay, kita bandingkan difference dulu. Point number 2, hang nampak ada satu biji glukos tu masuk dekat lengkuk tu. Itu point number 2. Okay, we can see that one glucose molecule or one amino acid molecule goes to the structure inside the carrier protein. So the name of the structure is active site. Nama lengkuk tersebut ialah active site. Okay. So, point number 2, one glucose molecule enters to the binding site, atau one glucose molecule enters the active site. Dua-dua betul binding site betul, active site betul. Okay, one glucose molecule. Macam mana saya tahu one? Saya nampak ada satu biji masuk dalam gambar tu. Okay. Okay ya. Nampak tak? Saya tak straight away cakap glucose diffuse from high region to low region. No. Kita cerita apa yang kita nampak dalam gambar dulu. Okay. Okay. One glucose molecule from outside the cell enters the active site atau enters the binding site of carrier protein. Okay, that is point number 2. Point number 3. Okay, point number 3. Okay, salin. Okay, saya ulang balik dari point number 1. The concentration of glucose outside the cell is higher than inside the cell. Point number 1. Point number 2, one glucose molecule from outside the cell enters the binding site of carrier protein. One glucose molecule from the outside the cell, one glucose molecule outside the cell enters the active site of carrier protein. That is point number 2. Point number 3, bila glukos dah masuk tu, apa jadi pada carrier protein? Carrier protein, seperti kawan kiri kanan kita ni sebut. Okay, carrier protein change shape. That is point number 3. Okay, carrier protein change shape. Ada orang dia gabung change shape, lepas tu glukos tu pergi ke to the other side. Betul, tak ada masalah. Okay, saya nak bagi key points untuk kawan-kawan yang tadi dia memang reaching out. Dia kata saya memang tak tahu cikgu macam mana nak jawab soalan begini. Okay, meh kita tolong. Okay, meh saya nak tolong. Okay, hari ni saya bagi apa key events for facilitated diffusion. Okay, mula kita bandingkan concentration. Nombor 2, kita habaq kata satu substance masuk ke binding site atau active site. Number 3, this causes carrier protein to change shape. Okay, point number 4, glucose molecule transported across plasma membrane. Tadi carrier protein dah change shape kan? So number 4, glucose molecule transported atau carried across the plasma membrane. Kalau nak sebut transported boleh, nak sebut diangkut boleh, carried tu diangkut kan? Okay. Transported across plasma membrane. Okay. So number 5 what happens after that? Bila dah transport, carrier protein goes back to its original shape. Okay, carrier protein goes back atau return to its original shape. Okay, so saya perasan bila saya browse through tadi, ada 4 orang yang bermula dengan dia starting betul, concentration of glucose outside the cell is higher than inside the cell. Lepas tu dia straight away terus berkata glucose diffuse atau glucose move from high concentration region to low concentration. Itu kalau kita nak cerita simple diffusion saya boleh terima. Sekarang ni kita cerita pasal facilitated diffusion, jalan dia lebih panjang. Jadi jalan dia mula-mula kamu dah identify mana high concentration region, mana low concentration region. Kita bagi nama pada region tersebut. Okay, kita tak boleh simply kata dah high concentration, low concentration region. Kita tengok gambar, kita tahu, high concentration region tu outside the cell. Low concentration region tu inside the cell. Jadi kita tak sebut dah dalam jawapan kita high concentration region. Kita dah tahu nama region tu apa. Okay, barulah kat situ menunjukkan kita apply definition. Kita bukan hafal definition. Okay, so cuba tengok lagi sekali. Number 1, the concentration of glucose outside the cell is higher than inside the cell. Okay. That is point number 1. Point number 2, kita cerita yang kita nampak pada gambar tu, one glucose molecule from outside the cell enters the binding site of carrier protein. Kita bukan simply kata glucose masuk dalam binding site saja. Kita kena habaq glucose tu mai dari mana, region mana. Kita tak boleh kata glucose from high concentration region. Mana satu? Luar sel ke dalam sel? Inside the cell ke outside the cell? Where does the glucose comes from? We have to mention glucose from outside the cell enters the binding site, because previously we have identified that the concentration of glucose outside the cell is higher than inside the cell. Jadi yang nak diangkut ialah kawasan luar masuk dalam. Sebab kawasan glukos luar lagi tinggi kepekatan dia daripada glukos kat dalam. Kenapa saya repeat part ni banyak kali? Sebab saya sedang tunjukkan jawapan yang tidak berubah walaupun saya repeat 3 kali. Jawapan dia tidak berubah walaupun saya dah repeat explanation ni 3 kali. Dia masih sama lagi. So jawapan yang saya tengah tunjuk dekat Google Meet sekarang, yang kiri sekali ni, jawapan dia belum berubah walaupun saya dah repeat 3 kali. Kita bukan sebut high concentration region, tapi kita terus identify high concentration region tu outside the cell. Low concentration region tu inside the cell. Dia tak ubah lagi ni dia punya jawapan. Saya assume internet connection dia tak berapa bagus kot hari ni. Okay. So sebab itu saya repeat 3 kali ya untuk anak-anak yang confused. Apa siapa cikgu? Repeat banyak kali ni cik dah faham dah. Kita ada kawan-kawan yang belum dapat lagi ya. Dia tak faham lagi. Sebab tu saya repeat dan saya cuba gunakan ayat yang berbeza. Kod ayat pertama dia tak dapat, ayat kedua dia faham. Ayat kedua dia tak faham, ayat ketiga baru dia faham. Sebab tu saya repeat 3 kali. Okay. Okay, saya nampak dah. Dia dah start tukar dah. Bagus. Okay. So, apa tadi? Kita dah identify difference in concentration. We said that one glucose molecule from outside the cell enters the binding site of carrier protein. Number 3, carrier protein change shape. Number 4, glucose molecule transported across plasma membrane. Number 5, carrier protein returns to the original shape. Okay, tadi saya sebut ada 6 points kan? So, point yang ke-6 ialah this process occurs until the concentration of glucose reaches dynamic equilibrium. Itu yang ditunjukkan dalam rajah yang saya tunjukkan tu. Molekul merah di luar sel adalah sama bilangan dia dengan dekat dalam sel. This process continues until the concentration of glucose reaches dynamic equilibrium. Untuk mereka yang menulis dari awal tadi, saya perhatikan dia punya evolve, dia punya advancement, dia punya improvement pada jawapan dia. Alhamdulillah, most of you got the 3 main point correct. Okay, 3 main points, 3 basic points describing facilitated diffusion dengan betul. Okay, jangan kecil hati bila saya asyik ulang-ulang dia ni jangan kecil hati sebab saya bercerita, saya narrate. I narrated my explanations a few times because I can see some of you keep changing, keep upgrading, and then these changes, these upgrades that you make in your answer, equilibrium. Alah siannya sayangku tak tahu ejek equilibrium, nanti saya tulis. Okay, I keep on explaining because I can see that as I explain, the more you change, you upgrade your answer. So this motivates me to repeat one more time until you get the good answer. Okay. Tak semestinya ayat tu sama dengan saya. Tak semestinya ya. So as long as the key events are correct. First, difference in concentration. Second, binding to specific site or active site or binding site. Number 3, change shape. Carrier protein change shape. Number 4, substance move across. Number 5, carrier protein original shape. Number 6, dynamic equilibrium. However you want to present the statement atau the concept. Setengah jam saja recall seronok nampak. Macam ulang belajar. Okay. So, for facilitated diffusion. Okay. For facilitated diffusion, tak apa tulis dulu. Saya tengah tunggu last kali ni. Siapa ni? Saya tengah tunggu Darwish update dia punya answer. Teruskan. Darwish teruskan. Last ayat. Last 30 second. Saya bagi last 30 second to update your answer. Equilibrium, saya tulis dekat dalam Google Meet chat ya. Equilibrium. Equilibrium. Okay. Okay, tak silent pula telefon ni. Ganggu ya. Alright. Okay. Saya dah lock screen. Sorry anak-anak, saya lock screen. So, saya nak tanya. Okay, pada slide ni, ada apa-apa lagi tak nak tanya pasal facilitated diffusion? Okay, what do you think? Apa-apa lagi nak tanya atau apa-apa komen pasal explanation tadi, ada setempat yang ada loophole lah, ada kekosongan, ada part yang tak ni. Okay. Okay. We all still stuck in this chapter. Semua orang tengah stuck dekat chapter ni. So don't worry. Okay, don't worry. Don't worry. Okay. Okay. Tak apa. Tak apa. Memang this kind of feedback memang it is expected starting from chapter 3. Because it is a totally new concept or terms. Masa kat chapter 3 dah baru kita kata, selamat datang ke biologi. Memang waktu ni lah. Selamat datang So, mungkin sebab kita tak buat amali lagi tau. So, bila kita buat amali, mungkin jadi lagi nampak kot. Because, itulah. Saya faham apa chapter ni. I've read research papers regarding this chapter lah. Orang academician peringkat universiti yang buat research pasal apa concerns or student concerns regarding this chapter. I think from what I read, it is mostly because the position of the chapter 3 ni betul-betul lepas chapter 2 dan saya pula terus bagi kamu jawab soalan yang macam SPM kan, dan kamu pula datang dari background yang tak ada PT3 last year. Okay, so saya faham, sebab itulah sejak dari awal lagi saya bagi soalan-soalan. Okay, ada soalan yang berkaitan dengan active transport kat sini. When ATP decompose into ADP and phosphate, what happens to the ATP? ADP dia memang berlegar-legar. It is abundantly available inside the cytoplasm. It is always ready near the ADP is always ready near the mitochondria, so that it can be turned back into ATP. Bila kita belajar fungsi mitochondria last chapter, we know that the function of mitochondria is to generate energy in the form of ATP. So mitochondria generate energy by connecting one phosphate group to the ADP. So tadi adenosine diphosphate, adenosine 2 kaki phosphate. Sekarang bila keluar daripada mitochondria jadi adenosine 3 kaki phosphate. Kita akan discuss lagi part ni dalam chapter cellular respiration. So, okay. Okay, tak apa, tulis whatever you are feeling. It is totally anonymous. And then I understand. Kalau tak saya memang saya berani jamin lah almost 50% of your questions for mid-year exam akan datang dari bab ni. Mostly dekat subtopik yang kita akan discuss hari ni, iaitu osmosis. Okay, so macam jangan buat conclusion dulu, suka tak suka. Kalau kita buat conclusion, suka tak suka, dia akan jadi barrier pada otak kita. Emotions akan menghalang kita daripada keeping an objective mind. Okay. Okay. Thank you so much for all of your responses. Confusing. Itu memang perkataan biasalah saya dengar sebab yalah, ya, saya faham. Okay, so tak apa. As we go through different questions, nanti kita akan cuba address lagi ya. Macam mana carrier protein yang glucose tadi tu change shape? Okay, carrier protein untuk passive transport, dia simply the presence of glucose inside the binding site or active site tu stimulate it to change shape. Okay, kehadiran glukos tu merangsang dia untuk mengubah bentuk. Untuk guna ATP, kegunaan ATP tu hanyalah untuk kalau kita nak lawan arus. Kalau pengangkutan tu nak lawan arus dari kepekatan rendah pergi ke kepekatan tinggi, kita nak paksa carrier protein tu change shape. Sebab tu kena ada tenaga. Tapi kalau simply follow concentration gradient, glukos ada, pop, terus tukar shape. Glukos datang kat binding site, pop, terus carrier protein tukar shape. Tapi sekarang ni dekat active transport, kisahnya ialah kita nak paksa carrier protein tu tukar shape ke arah bertentangan, ke arah low concentration gradient pergi ke high concentration gradient. So untuk memaksa pertukaran itu memerlukan tenaga. Okay, so we can see nanti bila kita discuss pasal ada syaitan pula kan kat sini. Okay, tak apa, it's okay. Okay, so I hope I answer your question. So sebenarnya anak-anak, apa yang saya sebut tadi? Banyak proses dalam badan kita yang start dengan facilitated diffusion tapi berakhir dengan carrier protein. For example, transport of glucose daripada usus kecil, daripada vilus pergi ke blood capillary. It started with facilitated diffusion, it ended with carrier protein because we want all glucose from our food enters our blood. Number one. Number two, another one is in kidney. We will learn later. The transport of glucose from kidney into our blood start with facilitated diffusion but ended with active transport sebab nak paksa. Okay, thank you so much for your responses. Okay, saya take note siapa yang rasa macam ada cakap confused apa semua ni, saya take note. Okay, it's okay. Saya take note dulu siapa. Okay. So, untuk part ni tak kejap ya. Untuk part ni saya yang akan explain sebab nak jimatkan masa, is already 9:44. Okay, tapi kalau kamu nak annotate pada kamu punya slide ni, kamu boleh annotate ya, tak ada masalah. Okay, kalau kamu nak annotate pun tak ada masalah. Okay ya. So, part ni saya akan explain. Satno, bagi saya masa 1 minit kejap nak prepare. Kejap ya. Okay. Sebab tadi saya punya tablet ni habis bateri. I want to draw using my tablet. So baru habis charge. Okay, I think I have the materials to prepare lah. Alright. So saya unlock screen pula. Alright, so I'm showing you my slide. Okay ya. So now let's see. Okay, the slide that I'm showing you right now is the sodium potassium pump that can be found in structure, in cells that can generate electrical impulse, macam nerve cell, macam muscle cell pun boleh. And then the difference in positive and negative charge tu yang akan menghasilkan electrical impulse. Tapi kita tak akan cerita kat sini. Kita hanya nak cerita pada level ini ialah how active transport occurs on the surface of nerve cell. Okay, kita belajar level 1 dulu. Level 1 dia ialah active transport in sekejap masa ni lah dia nak buat. Tadi dah try dah okey. Sekarang dah tak okey. Sekejap ya. Okay. So I cannot go to Google. Why I cannot go to Google? Ish. Okay, I cannot use this. Okay, tak apa. Okay anak-anak. Okay, macam biasa, whenever we start describing about transport of substances across plasma membrane, we start off with number 1, we start with difference in concentration. Okay, so kita bermula dengan cerita difference in concentration. Macam mana saya tahu difference in concentration? Cuba tengok pada gambar. Gambar ni dia label, nombor 1 dia label dekat mana extracellular fluid, dekat mana cytoplasm. That is number 1. So, based on the diagram, I can see that the concentration of sodium is the concentration of sodium ion is high in extracellular fluid compared to the cytoplasm. Nampak tak? Cuba tengok kat gambar pertama ni. Tengok gambar pertama. Okay, the concentration of Na plus is higher. Oh, besarnya Asad, no? Okay. Selalu lupa nak ijazah. Okay, the concentration of sodium ion outside the cell atau in extracellular fluid is higher compared to inside the cell. Okay, bila nak cerita transport, start dengan difference of concentration dulu. Why? Because usually we can observe the difference in concentration by observing the number of molecules atau the labeling inside the diagram. That is number 1. Okay. Number 2. Okay. Number 2, take a look at this. The movement of Na plus. So number 2 is how many movement of Na plus. So step number 2 is 3 Na plus bind to carrier protein. 3 Na plus bind to carrier protein. Okay, step number 2. Kena sebut berapa biji. Okay, kena sebut berapa biji. And then 3 Na plus bind to carrier protein. And step number 3, cuba tengok step number 3. Step number 3 occurs here. ATP decompose to become ADP. This is step number 3. ATP decompose to become ADP. Okay, gambar yang kedua tapi dah step number 3. Okay, gambar yang kedua tapi dah step number 3. Okay. Gambar yang kedua, gambar yang ketiga. Kalau saya cepat sangat, please stop me, okay? Okay, ini saya nak address isu beberapa beberapa kawan-kawan kita yang kurang jelas mengenai active transport. Okay, Hamim, Azlina, Darwish, Iman Safiah, sekiranya yang saya sebut ni adalah terlalu laju, tolong ingatkan saya. Okay, now that is step number 3. So, step number 4, hampa nampak tak kat sini? Bila ATP dah tukar jadi ADP P jauh, dah tak ada kat situ. ADP moves out of the picture. For step number 4, we can say that phosphate group binds to carrier protein. Phosphate group binds to carrier protein kat sini. Okay, dalam apa-apa tindak balas dalam sel kita, kalau perlukan tenaga, ATP kata hang nak tenaga. Okay, aku potong satu kaki fosfat, aku bagi kat hang. Lepas tu structure yang dapat fosfat tu akan bertenaga. Okay, nama dalam bio kita bagi istilah dia phosphorylated. Okay, I repeat, whenever ATP recognizes a structure inside the cell requires energy, ATP will decompose and give one phosphate to the structure. ATP has 3 phosphate. When ATP donate one phosphate to that particular structure, the structure will receive energy. It will be energized to conduct different processes. For example, for this part, ATP decompose, give one phosphate group to the carrier protein. Carrier protein energized to change shape. Okay? Itu yang ditunjukkan dalam nota hari tu, dalam video tu dia tulis phosphorylated. So phosphorylated tu basically bagi tenaga lah. Okay. So step number 4 sudah. Step number 5 is CP change shape. And then step number 6, kita habaq kata Na plus pergi mana? Na plus go where? It transported, Na plus transported out of the cell. Kan? Na plus transported out of the cell. Okay. So cerita Na plus dah habis. Number 1, concentration of sodium outside the cell is higher than inside the cell. Point number 1. Point number 2, 3 Na plus combine atau bind to the active site of carrier protein. Number 2. Number 3, ATP decompose to become ADP. Point number 4, phosphate group binds to carrier protein. Phosphate group provide energy for carrier protein to change shape. Number 5. Number 6, this causes sodium ion to be transported out of the cell. Okay, itu untuk transport of sodium. Saya nak tanya dulu, ada siapa-siapa nak tanya tak? Tengok dekat Google, tulis kat Google chat ke, unmute microphone ke, ada apa-apa nak tanya tak untuk part sodium ni? Okay, tak ada. Okay, dekat chat pun tak ada. Okay, sekarang ni kita tengok dekat potasium pula. So, potasium, potasium pun sama. Kita cerita dulu, step number 1, kita cerita dulu difference in concentration. Saya tukar teks warna lain. Nombor 1, kita cerita dulu difference in concentration. Saya tahu dalam buku teks, dia tak cerita pun difference in concentration tu. Tapi kenapa saya minta kamu tulis juga? Sebab it comes from our observation. It guides our observation. When we start with what we observe, it will guide our answer later. Kita sentiasa membiasakan diri kita observe dulu what what is the data that we have in the question ke, in the scenario ke. Kita sentiasa tengok, okay, apa data yang kita ada dan kita nyatakan data yang kita peroleh daripada kita punya observation. So from our observation, in the first picture, we can see that difference in concentration for potassium. Apa dia? So we can see that the concentration of K plus potassium ion is low outside the cell and high inside the cell. That is point number 1. Point number 2, berapa biji, okay, tengok dekat gambar nombor 4, berapa biji K plus yang masuk? Berapa biji? 2 biji K plus masuk kan? So step number 2, K movement of K plus. 2 potassium ions bind to the active site. Okay, itu step untuk K plus. How about phosphate? What happened to the phosphate? What happened to the phosphate? Phosphate leaves. Phosphate leaf carrier protein. And then when the phosphate leaf carrier protein, what happened to the carrier protein? Carrier protein change shape. Okay, carrier protein change shape. So when the carrier protein change shape, what happened at the final stage is step number 5. K plus go where? Alright, so potassium ion transported into the cell. Okay, so dia punya untuk K plus, the concentration of K plus is high inside the cell and low outside the cell. Number 1. Number 2, 2 K plus enters the carrier protein atau 2 potassium ion binds to the active site atau specific site of the carrier protein. Number 3, phosphate group leaves carrier protein. Number 4, this will cause carrier protein to change shape. And number 5, potassium ion transported into the cell. Okay. So I can see that most of you are writing several key concepts on your slides. Very good. Thank you so much for your attention. Okay. So hopefully we can understand the process of active transport better when we discuss it the second time. Tapi tak apa, if you still don't understand, there will be more discussions after this regarding active transport, sebab semua movement of substances that we discuss in this topic, it will be reused, will be rediscussed in other biological processes that occurs in our body. Memang kita akan jumpa lagi. Sebab itulah movement of substances ni kita belajar awal-awal, dan maybe some of you still didn't get it because you cannot see the bigger picture. Dekat organ mana guna, dekat physiological system mana guna dan sebagainya. It's okay. Bertenang dulu. Kita go through this sama-sama, kita cuba memahami sebaik mungkin. Mungkin bila saya bagi konteks lain, bagi keadaan lain, kamu jadi lebih faham. Ada setengah orang dia begitu ya. So don't worry. Okay, hari ni saya nak cuba masuk osmosis. So untuk osmosis, okay ya. Sekejap. Wait. We are supposed 10 to 10:30, we are supposed to have tutorial. Okay, we are supposed to have tutorial. But since tomorrow I will be on duty the whole morning, 7.30 sampai 9 esok tu saya memang tak dapat masuk. So, saya cuba bagi pengenalan kepada osmosis pada hari ini. The whole day tomorrow, 7:30 to 9 is your tutorial session where I will give you all sample questions regarding this particular chapter. Dari simple diffusion, facilitated diffusion, active transport sampai ke osmosis. Okay? So kita cuba masuk osmosis. Lepas tu kita tengok pula macam mana nanti lepas tutorial session ya. Alright. So what happens during osmosis? What happens during osmosis? Okay, so ini dia punya steps. 1, 2, 3, 4. But I will show you the steps here. Okay, number 1, semi-permeable membrane is impermeable to large molecule. Kalau kita tengok kat sini, molekul-molekul ni, molekul segitiga ni very large compared to the pores between the semi-permeable membrane. Line orange ni adalah semi-permeable membrane. So the triangles are bigger than the pores, number 1. Number 2, we can see that all of these triangles have water molecules attached to them because these molecules, these large molecules are can dissolve in water. Okay, so untuk menceritakan proses osmosis, number 1, we know that between solution on the left, kiri solution A, kanan solution B. So solution A and solution B is separated by semi-permeable membrane. And semi-permeable membrane is impermeable to large molecule. Large molecule cannot go through semi-permeable membrane. So, siapa yang akan bergerak? Sebelum ni, we discuss about simple diffusion. If we use the principles of simple diffusion, the 3 large molecules on the right will go to the left. The 3 molecules in solution B will diffuse to solution A. But that cannot happen here because the molecules are too large. So instead of balancing large molecules, we balance out the number of water molecules. So we can see that solution A on the left has a higher water potential than solution B. Mana cikgu tahu? Cikgu kira number of free water molecules on the left, number of free water molecules on the right. The free water molecules on the left the number of free water molecules on the left is higher compared to the right. The number of free water molecules in solution A is higher than in solution B. So instead of balancing out the number of large molecules, we have to balance out number of water molecules. So let's see what happened. So we can see that 3 water molecules from solution A diffuse to solution B. This process continues until solution A and B achieve dynamic equilibrium. What is the meaning of that? The meaning is the number of free water molecules in solution A is the same as the number of free water molecules in solution B. So, up until now, for the mechanism of osmosis, ada soalan nak tanya tak? Dalam kamu punya, dalam Telegram, dalam Telegram, this particular slide, I've shared with you the animated GIF form. Saya dah kongsikan format animated GIF untuk kamu refer in the future. The same slide. Ada apa-apa nak tanya tak? Okay, kalau tak ada apa-apa. Okay. Oh, boleh Alright. So, kamu tak tanya, saya pun nak tanya. Okay. For the first 5 minutes, saya bagi 5 minit untuk kamu baca muka surat 58 dalam textbook, iaitu what do you understand about types of solution and their effects on the direction of water diffusion. Please take a look at page 58 in your textbook. Siapa yang tak ada page 58, I will open it for you. Satno. Okay, I give you nanti pukul 10.10 kita start discuss page 58 in your text.
Cikgu, macam ni nak buka yang
Buka
online textbook?
Oi, jauh sangat tu nak buka online textbook tu. Oh, baru tanyalah ni pula. Sabar je lah. Saya buka page dia. Satno, I will share the screen. But macam mana nak download online textbook tu saya tak boleh cerita hari ni. Jalan dia panjang. Saya share dulu saya punya kopi. Satno. Attack pula. A window. Okay, this one. Okay. So I'm showing you right now page 58. Okay, ni page 58. Kejap ya. Okay, I think this is the biggest size that I can get. Kejap ya. Amirul nampak tak page yang saya pamer ni? Okay, kalau nak perlu saya besarkan ya. Saya sekarang tengah screenshot so that
tak apa, ni pun okey.
Okay, bagus. Thank you for your feedback. Saya screenshot juga untuk kamu refer dalam Telegram. Nanti dalam Telegram nampak tak? Okay, I think this is the okay, betul. Okay, bagus. Page 58. Okay, this is the gist of page 58 I've shared with you inside the Telegram. Okay, kalau tak nampak dekat dalam picture, dalam share screen. Okay, I can see that some of you have already shared what they understand. Okay, very good. Thank you Alif, Alina. Siapa ni? Ammar Hafiz. And then siapa ni? Amna Najiha. Okay, good. Okay, Ami. Siapa lagi ni? Kasrina. Okay. Humaira. Alright. Can see that? Marsha. Marsha ni saya ingat sebab sama nama dengan best friend anak saya semalam. Anak saya baru pindah sekolah baru. Dah jam 4 pindah sekolah baru. Baru dah jam 4 dah attend 3 different schools. Punyalah excited. Dapat jumpa best friend baru nama Marsha. Sampai sekarang saya ingat nama Marsha. Mama, kita ada best friend baru. Nama dia Marsha. Alright. Very good. Wow, Alina dia memang tulis sepenuhnya apa yang dia baca ya. Dahsyat Alina. Okay, thank you so much Alina. I love your enthusiasm. Sekejap ya. Saya nak stop presenting. I want to share the specific tab. Alright. Okey anak-anak, saya bersetuju, sepenuh hati saya bersetuju dengan semua submission yang diberikan dekat sini. It's okay kalau kamu tahu dapat faham sikit-sikit pun tak apa. Okay, yang penting kat sini ialah from what I read, you know that the solutions, okay, we talk about osmosis when we are comparing two different solutions, isolated by a semi-permeable membrane. Kita cakap pasal osmosis bila kita nampak ada dua jenis larutan yang diasingkan oleh semi-permeable membrane. Okay, that is number one. Dan kita pun dah discuss dulu. Kalau tak ada semi-permeable membrane, osmosis tak jadi. Number one. Number two, okay, kita tambah lagi daripada perbincangan kita last week. Kita tahu bahawa the two different solutions can be described using the term hypotonic, hypertonic and isotonic. The three terms, hypotonic, hypertonic and isotonic, describes the concentration of the solutions. Okay, kita gunakan istilah hypotonic, hypertonic dan isotonic untuk menggambarkan kepekatan larutan tersebut. Kalau larutan tersebut ada banyak molekul air compared to the other one, we call it as hypotonic solution. Okay, sebab bila osmosis kita sentiasa compare kan solution A dengan solution B kan. So for example, if solution A is a solution that has a lot of water molecules, so we call it as hypotonic solution compared to solution B. But if solution A has lower number of water molecules, kalaulah larutan A tu bilangan molekul air dia sikit berbanding solution B, so solution A is hypertonic towards solution B when compared to solution B. But if solution A has the same number of water molecules with solution B, so we call it as isotonic solution. So semua yang kamu tulis pada slide ini anak-anak, saya setuju sepenuh hati. Yang saya dapat fahami bahawa kamu tahu ada 3 jenis solution. Sama ada solution tu ada banyak molekul air, hypotonic. Solution tu ada sikit molekul air, hypertonic. Solution tu sama banyak molekul air dengan solution lagi satu, isotonic. Okay. So, okay, I give you another 30 seconds. I give myself another 30 seconds to read your submissions before I go through assessment. Assessment, mesti ada assessment punya. Saya nak assess apa? Kamu actually faham tak? Lepas tu, movement of water macam mana? Kan, sekarang ni kita semua dah kenal hypotonic, hypertonic, isotonic tu apa. So we want to apply it in a situation. We want to apply it in a situation to explain the movement of water. Sorry ya sayang-sayangku, saya pergi ke slide seterusnya. Tak apa. All of you have given good understanding regarding the terms hypotonic, hypertonic, isotonic. Siapa lukis-lukis tu? Oh, dia marah. Kau jangan marah dulu, Sat, no? Okay, Sat. Okay, ini adalah pentaksiran saya yang terakhir untuk hari ini. Iaitu terakhir ke, Sat? Saya rasa boleh tak? Ada lagi satu. Okay, so based on this diagram, based on your understanding about hypotonic, hypertonic and isotonic, we know that the movement of water is always from solution with high number of water molecules to solution with low number of water molecules. So in this diagram, there are 3 different U-tube. Bukan YouTube video tapi tube berbentuk huruf U dan di tengah-tengahnya ada semi-permeable membrane. Dalam tube U ni, sebelah kiri solution lain, sebelah kanan solution lain, kepekatan lain dan dipisahkan oleh semi-permeable membrane. So, bolehkah kamu indicate the direction of osmosis in the following solution? Direction of osmosis ya, gunakan arrow saja. Hanya lukis arrow saja. Bukan lukis number of water level lepas tu. Bukan water level ya. Solution, arrow tu. Arrow tu patutnya dekat mana? Saya perasan ada seorang betul dah. Okay, ada seorang dah betul. Position arrow dia dekat semi-permeable membrane. Okay, untuk orang yang lukis, dia punya arrow tu di atas tube U, di bawah tube U. Ayak keluar dari radas ke? Bukan. Ayak tu keluar merentasi membran plasma kan? Ada satu yang betul ni, Sadno. Ada satu yang dia punya position betul. Okay. So, rajah yang saya sedang tunjukkan dalam Google Meet sekarang, okay, saya nak tunjukkan ini adalah contoh position arrow tu betul ya. So the direction should be on top of the partially permeable membrane. Arrow tu mestilah merentasi membran. Okay ya. So kedudukan arrow oranye ni betul. Arah dia saya tak kata masih lagi tak betul. Arah dia masih lagi tak betul tapi position betul. Okay. Ada gambar yang kedua ni pun. Okay. Alamak. Dia dah tutup dah. So, kalau kamu lukis arrow tu di luar, ada tak orang lukis? Okay. Macam ni kan? Okay. So, I clear dulu. Okay. So, this one is not, has to be improved. Okay. Jawapan yang saya tengah pamer ni, kena improve sebab arrow tu berada di luar semi-permeable membrane. So takkanlah water diffuse di awangan macam tu kan? Macam dekat dekat stesen angkasa lepas pula kan? Macam dekat ISS pula. Okay. So saya kongsikan dekat sini. Okay, saya nak kongsikan jawapan daripada Shakira Alia. Okay. Jawapan Shakira Alia ni saya sangat bersetuju dengan jawapan Shakira Alia. Untuk tube yang pertama, Shakira tunjukkan the direction of osmosis atau the direction of water movement is from hypotonic pergi hypertonic. Bagus Shakira. Bagi tanda right pada tube U yang pertama. Okay. Pada tube U yang kedua, okay, sebab kedua-dua solution ni adalah isotonik. So bila isotonik, arrow pergi ke kiri adalah bilangan dia sama dengan arrow pergi ke kanan. Bagus Shakira, tanda right. Okay, and then kita tengok labeling saja sayangku. So arrow yang kanan sekali ni pun saya betul setuju dengan Shakira. Arrow that shows the movement of water is from the right to the left. Daripada hypotonic pergi ke hypertonic. Okay, kenapa jawapan Shakira ni betul? Sebabnya Shakira memahami bahawa the movement of water molecules is from a region that has a lot of water molecules to a region that has low number of water molecules. Explanation 1. Explanation 2, kita boleh juga kata Shakira memahami bahawa the movement of water molecules is from a region atau is from solution that has high water potential to low water potential. So, solution yang hypotonic has high water potential. Solution that is hypertonic has low water potential. Okay, so semua orang boleh refer pada screen Google Meet untuk tengok arrow. Okay, ramai yang sama jawapan macam Syakira ni. Cuma saya nampak dulu kan. Okay, so okay, okay. Tandakan jawapan masing-masing if you are correct. So I want you to know that um that this is the basis for osmosis. Number one, we know that when we talk about osmosis, we talk about the movement of water molecules. Number two, osmosis occurs because the solutions are divided or isolated by a semi-permeable membrane. That's why only water molecules move. Number three, I want you to identify, hypertonic solution like this, hypertonic solution like this, isotonic solutions like this. Okay, and then number four, I also want you to identify the movement of water molecules is from solution that has high water potential, has a lot of water molecules, to the solution that has low water potential or low number of water molecules seperti yang saya tunjuk dalam Google Meet. So ini adalah representation dalam makmal. What happens, apa yang berlaku bila kita kat mana kita nak apply pada badan kita. So pada badan kita, the difference in concentration of solution, difference hypertonic ke dan sebagainya ni ialah the two different solutions can refer to extracellular fluid atau bendalir yang berada di luar sel dan bendalir yang berada dalam sel iaitu cytoplasm. Semi-permeable membrane dia apa? Plasma membrane lah. So I repeat, in the diagram that we did before, we have two different solutions separated by a semi-permeable membrane. Dalam konteks of our body, within the context in our body, dalam konteks badan kita, dua solution yang berbeza tu ialah solution atau bendalir di luar sel dan bendalir di dalam sel diasingkan oleh membran plasma. So when we say hypotonic solution, isotonic solution, hypertonic solution, we always refer to the solution outside the cell. So for example, in the first picture, the solution outside the cell is hypotonic compared to cytoplasm. The solution, the middle picture, the solution outside the cell is isotonic compared to cytoplasm. Picture number 3, the solution outside the cell is hypertonic compared to cytoplasm. Gitu ya. So, saya berhenti takat ini dahulu pengenalan kepada hypotonic, isotonic and hypertonic solution. During our next class, I will provide you with for tomorrow's class, I will provide you with different situations and different questions so that you can apply what we have discussed today within the context of application of passive transport, active transport and osmosis in real life. Osmosis tu mungkin saya bagi soalan sikit je lah sebab kita baru belajar pengenalan. So anak-anak, basically this is the end of our class. Okay, this is the end of our class. Sebelum ni saya banyak bagi reflection on today's activities. Tapi sebab topik ni quite tough for you, I will give you a different type of exit ticket atau different type of reflection. Okay. So, based on your learning experience today, berdasarkan apa yang kamu telah lalui hari ini, okay, can you answer this particular slide? What I know is what you know before the start of our lesson. What I want to know is what are the things you still don't understand up until now. What I learned is what you already understand throughout the lesson. What I know ialah pengetahuan sebelum kita mula kelas tadi. Tapi yang tengah-tengah ialah apa lagi part yang kamu masih lagi nak tahu. What I learned, apa yang kamu dah belajar pada hari ini. Okay, so daripada kamu punya submission, saya akan identify apa lagi gap-gap yang perlu saya bantu untuk melengkapkan lagi pengetahuan kamu. Okay, based on your responses, I will try to identify what are the gaps in your current knowledge right now, and I will try to design our next lesson that can address this issue, that can address the gap. Okay. Okay, ramai lagi belum jawab ni. Siapa lagi ni? Okay, Dahia, Akma, Farah Kristina, Saidatul, Darwish. Okay. Akma Asfa. Ramai lagi ni. Okay. What do you think? Okay, what you want to know? Ada tak lagi persoalan-persoalan? Okay. Persoalan-persoalan. Okay. So, boleh tulis apa-apa pun. Boleh tulis apa-apa pun. Tak semestinya dalam konteks hari ni saja. Untuk what I want to know tu, mungkin macam what I want to know tu lebih kepada ada part yang macam masa discuss active transport tadi, ada part yang kamu masih tertanya-tanya, tulis kat situ. Okay. Okay. So ada seorang tulis kat sini, saya tak tahu nak jawab apa sebab saya tak faham sangat. Tak faham tu tak faham content ke, tak faham explanation pada topik ke, tak faham soalan tu tanya apa? Ya, yeah. So tak apa. Saya berharap saya cuba nak latih kamu to not understand. Kalau tak faham tu adalah human nature of being a student. Saya faham. Tapi a good student, to be a better learner, to be a better student, bila kamu tak faham tu, kamu boleh pinpoint kat mana yang kamu tak faham sebenarnya. So that is a characteristic of a good learner that can survive 21st century learning. Seriously. Sebab ilmu sentiasa berubah. But if you can understand the gaps in your knowledge, part mana yang kamu tak faham, kamu boleh pinpoint, that is the best. Okay, that is how you improve. So thank you so much for coming today. It's already 10:30. Thank you so much for all of your submissions. I really appreciate your attendance today, and thank you so much for coming. Tomorrow will be asynchronous learning where I will give you a student-based PDAC for you to answer. Thank you so much for coming. May Allah bless you. Have a nice day. Assalamualaikum.
Thank you,
Thank
Cikgu.
you, teacher.